All right, so microorganisms, starting off with bacteria. So bacteria are single-celled organisms. Their cell walls are actually consistent and composed of what's called peptoglycan. Peptoglycan is a mucopolysaccharide. Um, so these bacteria, they vary depending upon what bacterium you're looking at. They vary in different sizes and shapes and are classified differently. So, um, when we classify bacteria according to shape, then there's three different classifications. There's spherical or ovoid, and those are what's called cocci. Now, cocci are then subclassified. So if the cocci is occurring in pairs, then it's diplococci. If they're occurring in clusters, then it's staphylococci. And if they're occurring in chains, then it's strep. So staph occurring in pairs, it just sort of looks like two grapes sitting next to each other. When we are looking at staph, you're looking for like the whole grape cluster being together. And then when we're talking about streptococci, you're looking at, if you took all those grapes off the vine and you laid them all straight out, yeah, that's what it would look like. Then we have rod-shaped. So when we talk about rod-shaped, they are they're one organism, yeah, that is elongated. Yeah, and so it looks like you may have taken that grape and sort of squished it and then made like a hot dog shape out of it. Then we have spirochetes. Now, spirochetes look like a wine cork. So they're corkscrew shape. However, when you see the spirochetes microscopically, you have to keep in mind that you're not going to see that circular type um, visual because it's going to be cut in half. So you're going to see sort of a zigzag stripe happening. It's going to be flat. Now yeah, we also classify, the second type of classifications is how they stain. So your gram-positive bacterium are going to stain a deep purpley blue color. Your gram-negative bacteria are going to stain red in most techniques. So in some techniques, they are going to not absorb or remain unstained from the purple stain that you're going to give to it, and then they're going to absorb the counter stain, which is red in most cases. Then we can sort of further differentiate these bacterium into acid fasts or non-acid fasts. Yeah, so when we talk about an acid fast stain, we are talking about tuberculosis. When we are talking about non-acid fast stains, we are talking about bacterial stains and um, fungal stains. Now, gram stains are the stain that allows the organisms to be classified right off the bat into your gram positives and your gram negatives. Then we have fungi. So fungi are unicellular or multicellular plant forms, believe it or not. And they are a primitive plant form that have a nucleus, and they do contain genetic material. So their cell walls are composed of what's called chitin. So chitin, another example of chitin, would be like a crab shell. That crab shell, that hard exterior shell on the crab is chitin. So, if you think in terms of these little fungi, yeah, the outside wall that you're going to see microscopically is is a hard shell. It's that chitin. So we classify fungi just like we classify bacteria. And the first classification is called filamentous fungus, also known as hyphae. And they have a septum. So if we're looking at this, so this is the fungus. So this cell wall out here is that chitin material. It's a little kind of crunchy material, but it's not crunchy enough to nick your blade, so to speak, because it's so very, very microscopic. And then whenever you see this long fungi and it has these little sort of cross striations happening, those are what's called septa. And so any time that you see this septa, it's an automatic given that this is a mold type filamentous fungus. Then we have yeasts. So yeasts are budding. Yeah, they separate just like normal cells do. So you can see this cell right here. It's starting to pop apart. There's its genetic material here, and there's its genetic material right there. So they do the same thing. They push their genetic material to the edge. They start to, form that wall in between, and then they pop apart. So you can see this one here is just about ready to pop apart. Here's its genetic material. Here's its genetic material. And given about another day, this would become two fungi, which are then going to continue to replicate like that. That replication is called budding. Okay. Then we have yeast-like fungi. Now, the reason it's called yeast-like fungi is because it does reproduce like the budding fungus does, except that they elongate when they replicate. So rather than, two little circular cells popping apart like this guy's just going to elongate out, and it's going to make another long guy, and it's going to make another long guy. Yeah, and they don't pop apart. That's why they call them yeast-like. Yeah, so these guys do not have a septa. Yeah, so that's the way that you can determine whether it's yeast-like fungus or whether it's actually your filamentous fungus, because your yeast-like fungus will not have these little septa. Oops, I'm going the wrong way. Okay. Then we have protozoas. Yeah, protozoas are classified by how they move. They are an amoeba. Yeah, so they move by the extension of their cytoplasm. And that's called cilia or flagella. And they move their little cytoplasm or their little stem out here, their cytoplasmic extension, to move around. So look at this. This is our list of microorganism stains. However, you can see here, I've grouped them. So here are all of your AFB stains. That's your acid-fast bacillus. That's your tuberculosis and or leprosy. Then you have your brown and hops, your brown and bran. Yeah. So the brown and bran came first. Yeah. And so a guy named Brown and a guy named Bran got together and they built this stain. Okay. But then Brown got together with a guy named Hops and they built this stain. Yeah, they're both gram positive, gram negative stains. Yeah, but the the brown and hops is the derivative from the brown and bran. And the brown and hops is much easier to perform than the brown and bran is. The Kenyon and the Zeal Nielsen, these are exactly the same with one exception, and that is that the red stain, which is called carbolfuchsin, has a different makeup in between the two. Then the fights for leprosy, you're gonna coat, you gotta coat that leprosy bug. um, And it's the coating that we give to the leprosy that is going to actually stain. Then you have a fluorescent stain, auramine, rhodamine, yeah, for AFB. Up here you have two different Helicobacter pylori stains. You have a Diffquick and you have an Alcyon yellow, toluene blue. And those are specific for H. pylori, which is a very specific bacteria. Then you have a bunch of fungal stains. Yeah, so we've got the chromic acid shifts, yeah, which is just like your periodic acid, except that you're going to coat that fungus with the chromic acid, oxidize it out, and instead of periodic acid, but it's the exact same protocol, just with chromic acid as opposed to periodic acid. Then you have a Gridley, you have the GMS. Now the GMS is very close to the PAMS. And look, oh my, a Mayer's mucicarmine. Your Mayer mucicarmine, if you have yeast-like fungus, will stain that yeast. Then you have three different spirochete stains. You have the Worth and Starry, the Dieterle, and the Steiner and Steiner. They are all very, very close to one another. Yeah, the Worth and Starry is the go-to of these. Steiner and Steiner, yeah, did develop a spirochete. Dieterle did develop a spirochete. But that Worth and Starry is sort of the go-to. So we're going to talk about the Zeal, Nielsen, and Kenyon first. So it's the same stain. The only difference is the makeup of that carbolfuchsin. Now, when you do microorganism stains, you guys are going to have to bring in distilled water. Yeah, bring in yourself a good gallon of distilled water. Because tap water actually has dead AFB in it. And I know, right? Things that make you go, histology will change the way you view the world. But remember that it's dead AFB, so it can't harm anybody, but it's prevalent in a lot of water sources, and that's tap water sources. So it can, in fact, give your patient a false positive result, hence the reason for distilled water. Now, when we talk microorganisms, in any microorganism stain, your counter stain is very critical because we're talking about microorganisms, right? They're very, very small. Your counter stain can lay over those microorganisms, and it can hide those microorganisms, leading to a false negative result. So you have to be careful to make sure that your counterstains are not too heavy. Now, with a lot of the microorganism stains, some labs will choose to not only run a positive control to prove that their stain worked, but they will also use a negative control to prove that there isn't any contamination throughout the stain. So the negative control has to be cut on the same water bath on the same day as the patient does. And this is so that we can sort of prove that the water bath itself wasn't contaminated, and that our our water bath wasn't contaminated, and our stain line wasn't contaminated. Most of the time when we talk about a negative control, yeah, we're going to use a uterus, from a previously diagnosed case that does not have any mycobacterium in it. Yeah, so we're talking more like using a fibroid. Yeah, that, it's a known tissue type that does not carry bacterium. Yeah, so you would just cut that with your patient on your water bath. Yeah, grab your positive control that we know is positive, run your negative, run your patient, and that positive all together in the same batch. Now, with carbofuchsin, which is the actual uh, AFB dye, it's going to get this sheen over the top of it pretty quickly once it's exposed to air and light. So if you're flooding your slides, you sort of need to keep that carbofuchsin in motion. You have to keep moving that slide to prevent this very thick precipitation that can hang onto your slide, and it cannot be removed. So if it were me running my AFBs, I would use a Copeland jar, of that carbofuchsin, and put as many slides as I can in it. And I used to use my carbofuchsin for a couple of days. Yeah, so as long as at the end of the day, you take that Copeland jar and you put a lid on it and you put it back in the dark. Now, there is a debate, and that debate says that once you stain your AFB with the carbofuchsin, that it cannot, no matter how much you try, be decolorized by acid alcohol. Um, Others say that it can. So I don't know. I've never really tried it. But I've never really wanted any reason to try to decolorize my AFB completely either. So keep that in mind. Yeah, if you happen to forget about your AFB, in that decolorization step, I don't know, I would wash it in water and I would look at it microscopically and see if the AFB are still stained. So our protocol for both the Kenyan and the Zeal Nielsen, remember the only difference is in the makeup of that carbofuchsin. Yeah, so we're going to depair and hydrate to distilled water. Okay, we're talking distilled water always because we're doing microorganisms. Then you're going to go into your carbofuchsin. This is the one that gets gloopy and attaches to your slide. If you're flooding your slide, you have to keep in motion. And it's red, and that's going to stain your AFB organisms red. Then you have a distilled water wash. This is to remove that gloopy, gloppy carbofuchsin. Now, Carson suggests that after you stain your AFB with that carbofuchsin, that the remainder of the stain, you can use tap water because you've already stained that AFB, right? So I don't know. For me, it's just easier to keep it in my mind. um, this is a microorganism stain. I need to use distilled water. Carson's going to bounce back and forth for you throughout your chapter, um, from tap water to distilled water, from tap water to distilled water. Yeah, I feel like that bouncing back and forth makes it confusing. So I simply just use distilled water all the way through. Okay, if you want to take your chances, you go for it. But uh, in my mind, if it's microorganisms, it's distilled water constantly. So this is going to take off that, this distilled water, first distilled water wash, is going to take off that gloopy, gloppy carbofuchsin. But then you have to differentiate out that carbofuchsin out of everything else, other than your AFB. So we do that with an acid alcohol. Yeah, this is referred to as differentiating or decolorizing. And it's going to, take that red out of your tissue and leave behind sort of this really light pink faint staining. But your AFB that's there, your acid fast bacillus, is going to remain red. Then you're going to water wash to stop that differentiation from happening. And you're going to counter stain with what's called a methylene blue. Methylene blue is much lighter than aniline blue. And it will stain everything else other than your AFB a nice light sky blue. You should not be a deep dark blue. So if your counter stain turns out to be dark and your AFB are purple at this point, then you've overlaid your AFB with your methylene blue. And you need to drop it in this distilled water wash, and leave it there for quite some time to rinse out some of that methylene blue, to get it off of, layering over the top of that AFB. I always just really do this quickly just because our background, that's exactly what it is a background stain. And we just want to bring it up a little bit with colorization. We're not trying to deepen that blue into the tissue. Then you're just simply going to normal DCM. And get it? It's a red bug. I think that's cute. So your AFB is going to be red like the bug. And your background is going to be blue. This is what it looks like. So all these little teeny stripy red things here is AFB. And then your background is just, a light blue. This is another example. When I say you have to go bug hunting, it means that you need to be on high power and look for the microorganisms. So in this particular field, we have one microorganism right here, we have one microorganism right here, and we have one microorganism right here. Now, look at these guys versus these guys. This is how they should look, is that bright red. On the right-hand side, we have our microorganisms are sort of, purpley color, okay? This methylene blue, what laid over the top of that carbofuchsin. So our goal is to make them stand out bright red and lessen that background stain while still giving it a background to sort of pull this red up to the surface. Then we have the fights. So the fight stain, is for leprosy. And the leprosy organism itself, will decolorize after we have stained it. So what we're going to do is we are going to coat those leprosy bugs with a peanut oil. Now, the reason that we coat with this peanut oil is because when we do then go to decolorize, the oil is going to stick to that carbofuchsin, and it won't decolorize. It will remain red. Now, the trick with this is that you never, ever hit a fight stain with straight xylene. So when you depair and you go to hydrate, you're going to go into a peanut oil xylene mixture. If you go all the way to water, with your fight stain, your bugs are not going to stain, okay? So the only difference, if you look from here all the way down to here, it's an AFB stain, okay? It's an AFB stain with just a few tweaks to it. So we're going to depair in peanut oil xylene. And we're going to go into 2 changes of it. You're not going to hit it with any alcohols at all. So 2 changes of peanut oil xylene, and it's a 50-50 mix. Then you're going to drain your slides. So, oops, there's a weird 9 right there. So again, how I do it is I just pull them out. I lean them up against the side of the hood. I let it drain off, and sort of dry. And you're going to have leftover oil coating your section. So after I've given it about 5 minutes worth of drying, draining, then I take a chem wipe and I wipe off the slide itself of the oil, and then I lay a chem wipe down, and drop my slide section on you're blotting it basically, right? So drop your section down on the chem wipe, leave it there for a little bit, to let that oil sort of come off the surface of your tissue. Then you're going to go straight into that carbofuchsin stain. If you're flooding, keep it in motion. If you're in a Copeland jar, you can just drop it in the Copeland jar. Then you're going to come out and you're going to water wash it. That's going to remove that excess carbofuchsin. Again, this to this is an AFB stain. So you're going to come out and you're going to differentiate in an acid alcohol. The fights tends to differentiate. The differentiation is a little bit harder. I like to differentiate because I like to see it happening. I like to have a Copeland jar and I'll dip in, and you'll see that some of it will differentiate, some of it won't, and that's the oil that's still left in. So then I'll sort of keep dipping it until those red blotches of oil have decolorized. Then you're going to distill water wash That's going to stop that differentiation from happening. You're going to counterstain with that methylene blue, just like you did with the other AFBs. Then you're going to distill water wash to remove that methylene blue. This is a, a deviation from the AFB. So you're simply going to blot your slides like you did up here and let them air dry completely again on the inside of the hood. Just prop them up, let them dry. Yeah, and then you're going to mount normally, but you are never going to go through alcohol or xylene. You're never going to go through an alcohol or xylene up here, only your peanut oil xylene mixture. Now, when you mount your AFB, this is your normal, mounting media. You're going to have to use just a little bit more media because you're not going to have that xylene to help that media spread. And your results. Your AFB is going to be red, and you're going to have a light blue background. So this is what it looks like. This looks like there's nothing in it, but that is not the case. If you look at these little clusters right here, and you can see some little clusters in here and here, this is low power. But those bugs are definitely in there. So when you get on high power, it looks something more like this. The leprosy bug tends to want to grow in colonies. And so that's what you're seeing are these patches of colonies of that leprosy bug. So now we're flipping. Yeah, we're flipping over to gram positive, gram negatives. Yeah, so your brown and hops. Yeah, your brown and hops is a derivative of the brown and bren. Both of those are a derivative of the gram stain. So gram stains are for smears. And the brown and hops and the brown and bran are for are derivatives for tissue. But that doesn't mean that the brown and hops won't work on a smear, okay? So when you guys bring in your smears, you're actually going to run a brown and hops rather than a gram stain. The gram stain itself is more for blood smears as opposed to cytology smears. So the crystal violet is used first. And then we go into that slide with an iodine. And these two together make a mordant lake. Okay. Now, after your crystal violet and your iodine, both your gram positive and your gram negatives are going to be stained. But your gram positive cells are thicker. So when we decolorize, they won't decolorize as easily as the gram negatives will. Yeah, so that's why even though we're staining both of them here, when we decolorize, we're actually going to pull out the gram negative colorization, leaving behind the gram positive colorization. Again, and then we're going to go back and we're going to stain those gram negatives. Again, Carson suggests that your picric acid should be nearly anhydrous. We don't use anhydrous picric acid anymore, because it's explosive. She suggests putting picric acid in a little watch glass and allowing it to evaporate Please don't do this. We are not going to dry out our picric acid. Okay. Now, the other thing about the brown and hops is if your patient is taking antibiotics, you will probably not be able to demonstrate any gram positives because those antibiotics interfere with the cell wall of the gram positive bacteria. But this really isn't our call to make. This is more of the pathologist's call to make. He or she may look at these and say, why, we suspect that there's gram positive there. Why are we not seeing it? And make that phone call to the doctor to see if that patient is on antibiotics. So our protocol, we are going to depair and hydrate to distilled water. And this is done all flooding. Okay. And it's really quick. Yeah. So you got to be prepared to be on the move constantly when you're running brown and hops. So you're going to depair and hydrate to distilled water. You're going to go into crystal violet. This is going to stain both those gram positive and those gram negative bacterium. But we're going to pull that gram negative later on. Then we're going to distill water wash. This is going to remove that excess crystal violet. You cannot hold here or it's going to pull your crystal violet. In fact, you really shouldn't hold anywhere through this because it's such a quick stain that you constantly have to be on the move with this. So set yourselves up for success as opposed to trying to hold in any one of these things. Then we're going to go into that iodine lake formation with the crystal violet. So the iodine is going to attach to the crystal violet. It's going to convert the crystal violet to a salt that has low solubility. And so what happens is that when we go to decolorize, because we've converted this into a salt, we have more control when we decolorize. Otherwise, you would decolorize really, really quickly, and you wouldn't have much control over that decolorization step. So that's why we say this is a lake formation of a mordant, okay? Because it's going to paper clip to this crystal violet. Then we're going to distill water wash. That's going to remove any excess iodine. Again, you're going to lightly blot with damp filter paper, okay? Your filter paper needs to be wet, not dry, because we don't want it to dry out. If you let it dry out in any way, shape, or form, your gram negatives will not stain, okay? So it's just a quick blot on wet, damp filter paper. Then you're going to decolorize, and you're going to decolorize with acetone. This is quick. You can see this happen. You just flood your slide, dump it, and get it into that water, okay? And this is going to pull the coloration out of your gram negative. So quick flood into water. Then you're going to go into your basic fuchsin stain. Basic fuchsin is a red color. Yeah, and you're going to stain those gram negative bacteria. Then you're going to distill water wash. That's going to remove any excess basic fuchsin. Then you're going to go into what's called Gallegos solution. So this is what we call a diff and fix. So it's going to differentiate that basic fuchsin, meaning it's going to pull all the basic fuchsin out of anything that is not gram negative bacteria. And it locks in the stain, the basic fuchsin, that is in that gram negative. So it's a diff and a fix, okay? Then we're gonna distill water wash. That's gonna stop that diff and fix. Now, this is where the protocol changes, okay? So yeah, if we are going Carson, we're going to do this. If we're going Luna, we're going to do this. However, Luna, meaning, okay, so this is the brown and hops. This is the brown and Bren. Okay, the Luna is the brown and Bren. The Carson is the brown and hops. This is more like what I used to do, was Luna. But then we flipped over to the brown and hops from the brown and Bren. This is brown and Bren, brown and hops. We switched over from the brown and hops, or from the brown and Bren to the brown and hops because of this guy, the tartrazine. The tartrazine actually gave us our background, our yellow background color. And then the Cellusolve was the differentiation of that tartrazine. It could be either ether, glycol, or an alcohol that would differentiate that tartrazine. But we used this tartrazine for a very, very long time until all of a sudden they decided that tartrazine was a carcinogen. And so we switched over to the brown and hops. So you guys are going to run the brown and hops, which means that from your distilled water after your diff and fix, you're going to go into acetone. And that's really, really quick, because all you're doing is introducing that acetone to the slide in anticipation for our stain of the background, because it's acetone based. So it's a picric acid acetone. And so you're going to introduce the acetone, go into the picric acid acetone, and that's quick as well. And the picric acid is what's going to give you that light yellow background stain. Then we're going to go into another change of acetone. This again is quick. And it's going to diff out the picric acid that may be laying over any of your bugs. Okay. So you're going to go into acetone, you're going to go into acetone xylene. So you're continuing to diff, but you're also starting to clear. And this is quick. And when I say quick, I mean quick. So it's like dip, 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 and boom, you're going to stop that diff by going into straight xylene and then another straight xylene. I will stop in this xylene and I'll soak my slide. Just to make sure that I've actually stopped all of that acetone from differentiating my slide out. And then you mount. So your gram positives are going to be blue. Your gram negatives are going to be red. Your background is going to be faint to no yellow. And your nuclei are going to be light red to pink. And this is what it looks like. So here we have colonies of the gram positives, and here we have colonies of the gram negatives. We also have some areas where they're sort of co-hosting together and mingling with each other. Now, the way that I remember which one is which is as simple as it is. I think of a stoplight. And at a stoplight or a stop sign, it means no, negative, you cannot go, right? So because it's negative no, that means that your gram negatives are red, like the stoplight or the stop sign. And then you can deduce from there that the purple is the positive. Now, over here on the right, we have colonies of our positives. And then we have sort of some sporadic negatives over here on this side. Then we have some, oh, yeah. Then we have Helicobacter pylori. This is referred to as H. pylori. Yeah, H. pylori is really very, very important to the patient. Yeah, the presence of this bug is because it's been found that H. pylori is associated with gastritis, peptic ulcers, and it increases the probability and risk factors for those to turn into carcinomas and lymphomas with the presence of this bug. Yeah, so when they it used to be years ago that, um, you know, ulcers and peptic ulcers and gastritis, inflammation of the gastric lining was all due to what the person ate until they discovered this bug. Now, we haven't known about this bug for very long. Yeah, we've probably known about this bug for maybe 25 years. Yeah, and maybe more like 28. Yeah, but that's a drop in the bucket of water compared to how long we've known about everything else. Yeah. So this bug was just discovered in that if the H. pylori is present in the small biopsies of colon and stomach, yeah, that patient is much more likely to have that area turn into a carcinoma or a lymphoma. So the Alcian yellow, yeah, you guys are familiar with Alcian blue, but this is Alcian yellow, combined with the T blue, yeah, and we stain it for H. pylori. Now, the Alcian yellow is very, very similar to the Alcian blue. It will stain mucins yellow. Yeah, the T blue will stain just like you previously saw in your connective tissue and muscles. Yeah. So our protocol, we're going to depair and hydrate to distilled water. We're going to oxidize in periodic acid. We're going to water wash to stop that oxidation. You see what I mean and then there's metabisulfite, right? That's that optional step to prevent that non-specific staining. You see what I mean when I say, the stains this quarter is harder, but it's also kind of easier because this to this is just the very beginning of a PAS, right? Yeah, and you're doing the same thing. You're oxidizing, water washing, preventing nonspecific staining, and then removing that metabisulfite. But here is where it changes. So you're going to go into the Alcian yellow. This is the mucin staining. So one might ask, why is it, that we want to stain the mucins? Well, the reason for that is because the H. pylori bug actually feeds off of mucins. So it likes to embed itself in the crypts and the folds of the mucins that have just spit out into from the mucosa out into the lining of the stomach and the lining of the colon. So So these bugs sort of conglomerate to where the mucins are being released out of the um, uh, 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 mucosa of the colon and the stomach. So by staining those mucins yellow, we can sort of see through that yellow mucin to actually see the H. pylori that are going to pick up the stain, the T blue stain. And so you're going to see these sort of uh, swimming little bugs inside of these mucinous deposits. So in order to do that, we're going to stain the mucin yellow first. Then we're going to water wash to remove all the excess alcian yellow. Then we're going to stain the bugs, the H. pylori, with T blue. Now that T blue is also going to stain the nuclei that's there. Yeah, then you're going to water wash to remove that excess T blue. Then you're going to blot it. Okay. So it's the same story, same song and dance, drop that slide down onto filter paper or onto a Kim wipe, and then pick it back up and dehydrate it quickly. And it's not necessarily the xylene. Once you hit xylene, you are fine. But it's that alcohol that may pull some of your T blue. So you're going to go through your alcohols quickly. Once you hit xylene, you're fine. And then you're going to mount. And then this is what it looks like. So yeah, your background will be like a light blue, maybe some little purple hues to it from your tea blue. Your mucins are going to be yellow. So here's that sort of mucin bath. And then all these little squiggly swimmer guys in here, that is the H. pylori, that's feeding off of the mucins. All right, then we have the Hodgkiss McManus for fungus. This one you already know. It's the PASG, okay? The only difference between the normal PAS and the PASG is one change. And that's because the PASG can also be referred to as the CAS stain and instead of the PAS stain. Because we have those fungal cell walls that are chitin, we have to use a harder acid to oxidize than the periodic acid. Now, with your CAS, you cannot use periodic acid, unlike the PAS, where you can choose to use the chromic acid to oxidize. The periodic acid is simply not strong enough to oxidize that chitin cell wall. So we have to use chromic acid. You can't interchange it from picric acid. Please be careful. Don't spill chromic acid on yourself. This is a true picture from the old lab on campus over in building 16, sorry, of a guy one of my students who dropped chromic acid on the floor, and this was the mess we had to clean up. Just saying. All right. So You're going to run it just like a PAS with one exception, and that would be chromic acid instead of periodic acid. And this is what it looks like. This is budding fungus inside of a kidney. And your pathologist may or may not want you to counter with that light green. Yeah, if they don't want to counter, this is what it looks like. And here are your fungus deposits. Then we have a gridley stain. This should be familiar as well. So basically, in a nutshell, the difference between this stain and the CAS is that after you stain the fungus with the shifts, so you've got chromic acid, okay, then you got shifts, then you're going to develop that shifts. You're going to increase the depth of that shifts by going over the top of it with an aldehyde fuchsin. Yeah, and then you're going to water wash, and then you're going to counter with our favorite mental yellow, but it's actually pronounced metanol yellow. But because it's hard to keep in, some people refer to it as mental yellow. Yeah, and then you're going to dehydrate, clear, and mount. So it's really PAS with chromic acid and then going over the top with an aldehyde fuchsin. Here's another one that you know. So it's the GMS. Now, the only difference between the PAMS that you have already learned and the GMS is that the periodic acid or the PAMS uses periodic acid. The Grocott methamphetamine silver uses chromic acid. Okay. Again, we're going after the bugs, right? The fungus with that chitin cell wall. So if you remember the fungus, it has chitin, yeah, and you have to oxidize it harder, then it should always bring to you the idea that it's not periodic acid, but rather that it's chromic acid because you've got to get through that chitin. Now, last but not least, we have the Werthin story. Every person has their nemesis tissue. Mine is thyroid, and every person has their nemesis stain. Mine is the Werthin story. You'll be happy to know that I have a nemesis. Yeah, and it has in fact been tested by a student almost every year. They don't believe me. Um, but I'm telling you that if I were in the lab with you and all I did was pick up your slide and move it from one Copeland jar to the other Copeland jar, it will not work. I have never been able to actually perform this stain. Yeah, I have done it identical, standing next to many a people who say, well, do it just like me and it will work. And I will say, okay. And I will do it identical to them with the same times, with the same water baths, with the same solutions. Mine will fail and theirs will work. It just is. And I don't know what it is, but it's got to have something to do with, because I couldn't do it in my lab either, and I couldn't do it in the student lab either. So it's got to have something to do with my chemical makeup or something that interferes. But there you have it. There's my downfall. So The Warthin Starry is what's called a argyrophilic stain. The opposite of argyrophilic is argentafin. You are going to start running into this all the time. So you need to know the difference between argyrophilic and argentaphilic. So an argyrophilic means that your target area, whatever it is that you are targeting, has the ability to bind to the silver. Because it has the ability to bind to silver, you do not have to reduce that silver. Okay. Um, that's not right. Our gyrophil is the other way around. Our gyrophil does the actual organism. They do not have the ability to reduce silver on their own to a visible color. I had it backwards. Um, so you have to use a chemical reducer to make it visible. Um, and the way that I caught that in my brain is from this right here, our gyrophil, right? So I looked up and I saw this and I went, oh, wait a minute, that's not right. And the reason this flashed my brain basically is because I remember an argyrophil stain by the fact that you have to fill another Copeland jar with a reducer. Okay. So our gyrophil is the extra step in there, the extra step of the reducer. Argentafin is when the target has the ability to bind to the silver and can reduce itself. Okay. So that's like the PAMS, where the Warthin Starry, you have a reducing step within it. Now, silver stains for bugs are considered more sensitive, especially when we're talking about just a few little guys in your section. So that's why, yeah, when we are talking about the GMS, if we only have, say, 3 little strands of fungal organisms in there, they are going to show up better with the GMS than they would if we did a PASG on it or a CAS. So when we are using the reducing substances, the reducing substances are also going to stain silver. So if we as histotechs have, formed, gosh, formalin pigment has occurred, okay? That formalin pigment is also going to stain with the silver. Yeah, and formalin pigment is considered a reducing substance. So it's going to grab a hold, and there are more reducing substances, and we'll get there in a couple chapters. But for now, formalin pigment is really the only reducing substance that you have encountered. So if you run your Warthin Starry, and your pigments have shown up, and they look black like silver, like a silver impregnation would, don't worry, just sort of look past it, to go look for those spirochetes. All right, so The worth and story. Before you even begin, you need to have a 2% silver and a 5% gelatin and a hydroquinone solution in separate plastic tubes, or in Copeland jars, okay? And sink them into the water bath and heat them up for 1 hour, until they're like 54 degrees. Now, here's the thing. You can't test that with a thermometer, okay? Because there's metal and a thermometer. So you just have to heat it up, basically. But if you get asked this question on your certification exam, you'll need to know that it's 54 degrees, okay? Then you're going to get yourself a graduated cylinder and a Copeland jar, a chemically clean one, and you're going to put them in the oven at low heat. You're going to need those later when we develop the slide. So because you're using these heated solutions, you have to heat your graduated cylinder and your Copeland jar, or they're going to crack your Copeland jar. So you get all these guys heating up. Then you depair and hydrate your slide to acidulated water, okay? Where right off the bat, we are going to provide an acid environment. So this is distilled water with citric acid in it. You should have a pH of about 4, and you can check that with the pH strip. If it's not, you just put in a couple more drops of acid until you are around 4. Then you're going to, wash your slide in your acidulated water, and you're going to go into a 1% silver solution, impregnation solution, and drop it into the water bath. Okay. This is going to impregnate, but while you're impregnating it, yeah, and we're talking, Copeland jar, right? So while you're impregnating that slide, that's when you are going to take all of this and you are going to prepare that developer solution. And you can follow Carson on how to mix that. Then you're going to place it into the water bath. And then you have this developing solution as well. Yeah. And so this developing solution, that you've mixed here, you've placed it, you've used these guys. Okay. You have mixed your developer. Then you now have the developing solution, that you're going to pour into, a Copeland jar. You're going to put that in the water bath along with this guy. So now you only have 2 things in your water bath. You have your slides that are in the 1% silver solution impregnating. Now you have a warm developing solution in the water bath. You're going to pull your slide, and you're going to remove this excess silver, and you are going to drop it right into the reducer. Then you're going to pull it, distill water wash to remove that excess silver and developer, and DCM. So you're just going to, this right here is sort of the hard part, okay? While the stain itself is really simple, okay? Yeah, you're just going to impregnate, you're going to develop, you're going to wash it away, and you're going to DCM. So this is what it looks like. Your spirochetes are black. If any other bacteria are there, they are also going to turn black. And then your background should be sort of this light brownish yellow, and that comes from that developing solution, okay? It actually comes from that hydroquinone. FYI, hydroquinone is a carcinogen, so make sure that you're using it under the hood. So all these little black guys are spirochetes. And if you look at this one down here, you can see that sort of zigzag form on him. And that's that corkscrew shape. Now, you guys have the ability to get right up close and personal with these spirochetes by putting it under the Vision Tech, and you should be able to really discern that sort of corkscrew, yeah, zigzag shape. So there are variations of the Warthin Starry. There's a Dieterle, a Steiner and Steiner, and there's a microwave method. And they're really just one difference from the Warthin Starry. So you can study these guys on your own. Should you decide to do the microwave method, I caution you, it goes really fast, and it flashes really quick. Um, so most people actually are still doing the Worth and Starry via water baths. Okay. Now this is what it looks like if I do it. Um, but this is what it's going to look like for you guys. Okay. The end.